Hello, everyone. My name is Silvia, and in the next 30 minutes, I'll talk about quantitative and especially the spatial differences in host response dynamics to SARS-CoV-2 and its variants of concern. So we've all experienced the last 2 years that SARS-CoV-2 has multiple variants that occur in recurring phases and waves. And the most prominent of these variants are, for example, the beta variant that emerged in South Africa, also known as B1 or 351, or the Delta variant, known as B1617.2, and also recently the variant B11529, also known as Omicron. And what we observed is that all these variants generate recurring waves of infection. So these variants have different, or let's say evolved transmissibility. They show different rates of infection, causing different rates of hospitalization of the patients. But what all these variants have in common is that despite their very common clinical manifestations in general, they have very different rates of infection in different patients. So we know by now that age and comorbidities, for example, are factors driving severe trajectories and symptoms. But we also have young patients that are perfectly healthy but still show severe symptoms when in COVID-19. And on the other hand, we have older patients with comorbidities that have asymptomatic course of infection. So we still don't know why this is the case. And there is a balance between two leading factors in that regard, which is the antiviral response versus the immune evasion. So antiviral responses are important for the body to fend off the virus, while on the other hand, the virus is trying to evade the host immune system. So this is something that we need to look into in further detail to see actually why certain patients are asymptomatic while others develop a severe disease trajectory. So let's start at the very beginning. SARS-CoV-2 is an airborne particle. So once it's inhaled, it infects cells of the respiratory system. So the primary infected cell usually replicates the virus and releases it again. So the cells neighboring the primary infected cell, also known as bystander cells, either experience emerging virus particles that were replicated previously from the primary infected cell, or they can also experience signaling, hence paracrine signaling, from the primary infected cell to develop some antiviral response. Let's look at this in more detail. So a cell infected by a virus has various pathways that it can activate to generate an early cytokine response, meaning that it produces interferons and TNF-alpha and so on. And these cytokines are released from the primary infected cell and more or less warn neighboring cells, hence bystander cells, in a paracrine fashion, so that these can actually mount an antiviral response. What does this mean? So interferon beta, for example, induces an antiviral response, which is best represented by ISGF3, for example. And this complex translocates into the nucleus and activates the expression of roughly 350 antiviral genes. For example, DDX58, which codes for RIG1, which is a cytoplasmic antiviral sensor. And once a cell has entered or expressed all these genes, it's said to have entered an antiviral state. So it's just better prepared to fend off any approaching virus particles. So it's really about the difference between the primary infected cells and the bystander cells that makes a difference to actually differentiate between how well a cell or the body in general is able to mount an antiviral response and to fend off a severe trajectory, for example, in COVID-19. Investigations on the differences between the infected cell and the bystander cells are already undergoing and have been described in some detail over the last 2 years. and for other viruses, of course, in the years before. But most of these studies use single-cell RNA sequencing, which is a very valuable tool, but to some extent it misses the spatial context. So if we have here in blue all the bystander cells and in yellow the infected cells, and we see that these groups separate, so we have different transcriptional responses, but we don't know which of these blue bystander cells actually sits next to an infected cell, so that it would experience the paracrine signaling directly. So here is where our spatial analysis comes in. We really want to know which bystander cells are sitting directly next to the infected cells, hence which cells can really experience the full extent of paracrine signaling. And throughout the talk, I will also focus on the heterogeneity that we observe in SARS-CoV-2 infected cell cultures, which kind of resembles the complex heterogeneity we see in the symptoms in patients. And I also want to focus on some signaling pathways that are affected by SARS-CoV-2 and its variants. So here's a short depiction of the workflow that we use in our lab. I want to start with the infection of the cells. So for that experiment, we use H-1BD slides that we infect on color-free cells, for example, and others that we infect with different variants. We incubate the virus for 1 hour and remove the virus again. We wash the sample, and then we incubate for another 24 hours to give the virus time to be replicated in the cells and the cells to mount responses and so on. So at the beginning, of course, we need to determine how much virus we need to properly infect our cells to gain an adequate infection rate. And here we measure our viral load in plaque-forming units, or PFU. And here you see immunohistochemistry targeting the nucleocapsid protein of SARS-CoV-2 after infection with different viral loads, ranging from 0.6 PFU to 600 PFU. So what we used for our final experiment was 30 PFU to not overload the cells, which we almost already observed at 60 PFU, but we still have enough infection to detect some reasonable infection rates. So everything that I will be talking from now on was done with 30 PFU infection. So we checked an effective infection using qPCR. Here you see that we have a CT value of about 15 in all our data, all our samples basically. And the infected area varies between the variants. So we see a lower infection rate in the Omicron sample than we do in Delta or Wuhan, for example. And here's the images that we use to calculate that data using Cell Profiler. So there's already some differences in the area these cells infected, but less so in replication, as represented by the infected area versus the qPCR, respectively. So we then move on to check our data using molecular cartography, which is basically a multiplexed single molecule fluorescence in situ hybridization technique. And it allows us to study antiviral responses of up to 100 genes simultaneously. So it basically works by hybridizing probes that are specific to each of our targets. You color the probes, and then you image the sample and you decolorize again. And if you repeat the process 8 times, you generate 8-bit code that is specific to each target. So here at the top, you see representative images of round 1 to 3 out of 8 of these imaging rounds and overlapping images. And sometimes these dots would show up or not, generating ones and zeros in the code. And as I just mentioned, these codes are specific for our targets. In the middle right part, you see the resolution we have. And if you look at the very right picture of these 3 bluish images, you see that we have a resolution below 1 micrometer. And this is really important if we want to check cells that are really loaded with virus or with antiviral responses. So sometimes responses are just too strong, so we can't decipher all the signals properly. So having high resolution is really important in this aspect. So we did our experiments first in biological duplicates, and here you see that the two slides we infected and analyzed show quite similar responses, which shows that our approaches are reproducible, which is also good and validates our data. So I want to show you a quick video on what the final data is like. Here we have in blue our nuclei, and in yellow, these bigger yellowish dots are SARS-CoV-2 nucleocapsid protein RNA particles that stand out in the midst of all the other transcripts. So all the smaller color dots are the transcripts we are actually targeting. Just to give you some imagination on what the data and the resolution looks like. So we tested SARS-CoV-2 infections in different cell lines, originating from pulmonary, colorectal, and hepatocellular cell lines, basically. And here you see an image of HH7 cells, which is a hepatocellular cell line. And in pink, you see SARS-CoV-2 particles. You see the primary infection site, primary infected cell, from which the SARS-CoV-2 signal radiates towards the surrounding cells. And we have multiple infection sites. You see the other pinkish areas in that image. So we have multiple individual infection sites that radiate the virus from their outbound, basically. We also did the same experiments in other cell lines. CAL-free cells is a pulmonary cell line, and CAL-2 cells is a colorectal cell line. And we see that the cells are differently capable of being infected by the virus. And CAL-free cells show the highest infection rates per cell, as shown here in D. We also checked whether virus infection actually induces up or down regulation of entry factors. ACE2, furin, and TMPRSS2 are currently the most discussed, or let's say widely discussed, entry factors necessary for SARS-CoV-2 to enter a cell. And we checked whether infection actually affects these, but we did not see any major changes regarding these, the regulation of these entry factors. Something else that was very interesting we observed was that SARS-CoV-2 nucleocapsid RNA particles tend to localize close to the nucleus, and we hypothesized that this is or indicates towards ongoing translation at the ER. Yeah, this is a really interesting finding. So we then moved on to studying infections of the different variants in CAL-free cells, since those nicely or are the best representative cells for the respiratory tract. And here you see a principal component analysis on the graph on the left, where you see in purple the non-infected cells. So all our replicates group quite nicely in the right bottom corner. And we have the different variants of concern here in bluish, green, and red. And you can see that the Omicron variants here shown in blue are kind of located the closest towards the mock culture. But we see that the infections, the different variants of concern, actually spread out across the PC2. So we were checking which genes are actually responsible for generating this difference on PC2, which are the graphs on the right. So we checked whether the genes that are important for the spread of the infected cells against the mock culture in SARS-CoV-2 are also important in the other variants. So here you see the three most influential genes in SARS-CoV-2 depicted in all the different variants. And you see that the NF-kappa B inhibitor alpha, which is represented in this red dots here, really nicely locates within those white areas. These are single molecule fish images, or basically approaches of SARS-CoV-2. So in white, we have our virus infection, hence highly infected cells. And in blue, we have deciphering. So those red dots really nicely co-locate with the white areas. And in some areas, let's say the really highly infected cells, we also see expression of interleukin 6, which is represented here in green. If we move to the right, which is the B16172 variant or Delta, we again see that highly infected cells represent or express a lot of NF-kappa B1 alpha, and also in Omicron, but to a lesser degree. So we also see a decrease in the expression of NF-kappa B inhibitor alpha. So here's just a statistic analysis as well. So highly infected cells do indeed express increased amounts of NF-kappa B inhibitor alpha, which led us to hypothesize that NF-kappa B inhibitor alpha might indeed be a good marker for highly infected cells in all variants of concern. And this might also hint at immune evasion, because NF-kappa B, as you remember before, is one of the main pathways in which cells use to mount an innate immune response. And high expression of its inhibitor, of course, might interfere with the process. So we also looked at the genes that are responsible for the differences of the infected cells towards the mock in the other variants. And here in the right top images, you see the C19orf66, which is an antiviral gene, and STAT2 and MUC1 represented on the images. These genes were responsible for the delta differences, or defining the loading scores in the PCA. And if you look at cell 1, for example, in the delta variant, you see a strongly infected cell here represented by a white big dot. And right next to it, you see another cell with a lower infection rate that has an increased amount of these bluish particles representing C19orf1. So this cell, the neighboring cell, actually induces or mounts a stronger antiviral response than the primary infected cell. If you look at the bottom two images, which is the OM variant and the mock variant, we see that CDX58 and IFID1, two other antiviral genes, are also highly expressed in primary infected cells, as you see here in cell 1, including CXCL10, which is a powerful chemokine, also important for immune attraction of other adaptive. So we checked the differential expression of genes following infection with these variants, and we found that a number of core genes is differentially regulated in all of these variants, leading by NF-kappa B1 alpha and other genes that I've already mentioned. And we've seen that, or as you can see here in the Venn diagram, we have a large number of genes that are conjointly regulated by SARS-CoV-2 and the Delta variant that are not so much differentially regulated by Omicron. So we already see that the original SARS-CoV-2 Wuhan strain and Delta are way more similar in their host immune responses, or in the host immune response that they trigger, than the Omicron variant. We further looked at single cell data. So here you see that the variants, hence SARS-CoV-2, Delta, and Omicron, represented in red, green, and blue, move away from the purple cell population. Here we tested roughly 25,000 cells. And from that cell population, we tested, or we divided basically the cells in bystander cells and infected cells based on the fluorescence intensity from the SARS-CoV-2 N plus particles. We saw that again, roughly 40 to 60% of cells were highly infected in Wuhan and Delta strain, whereas only about 30% were infected in the Omicron variant. On the right, in D, you see a representation of all the cells divided in infected and non-infected cells, and we see that overall we have the infected population shifting away from the bystander or non-infected population. So here we have UMAP maps from the different variants. And we see that in Wuhan, for example, we have a population shifting away from the rest of the cells, and we see the same in Delta. But looking at Omicron, we almost have the infected cells almost seem to blend into the non-infected population. So meaning that responses in cells infected with Omicron are not that severe or that different to the responses in MOX than we see than the difference we see in SARS-CoV-2 Wuhan. So having differentiated the cells between, or let's say, divided into bystander and infected cells, we again checked for differential expression. And we found, again, a large number, let's say 15 core genes that are differentially regulated in all these genes. And when we check for biological processes and pathways that are affected, we see that all of these responses correlate to antiviral signaling and response to cytokine and well, responses to virus infection, so to speak. So which was really interesting was when we checked the responses in bystander cells. So we see that there's a large number of differentially expressed genes in SARS-CoV-2 that are not differentially regulated in Delta and Omicron. So again, we checked what these genes, which pathways, which biological processes these genes might relate to. And again, we see a very strong relation to cytokine signaling and relation to immune signaling response to virus and so on. So we next checked which genes might correlate their expression in response to the virus. Here in the bottom triangles, you see the correlation of the infected cell population, and on the top triangle, you see the relation of genes in bystander cells. It was interesting that in infected cells, we saw that, let's say, a core set of genes were correlated in all different variants. Here we have Wuhan on the left, Delta in the middle, and Omicron on the right. But we did not really see correlation of genes in bystander cells. We have a small correlation of genes in the Wuhan strain, none in the Delta strain, but we see a strong correlation of antiviral genes that we already know from previous analysis, including JAK-STAT signaling that correlates in bystander cells. So here we have STAT1, STAT2, and EphA1 and EphA1, which are downstream activated genes of JAK-STAT signaling of ISGF3 that I've been mentioning before. So we see that infected cells kind of tend to show similar responses, while the bystander cells exhibit different expressions and also involve JAK-STAT signaling in the Omicron variant, which is not so obvious in the other variants. And if you remember that I mentioned that JAK-STAT signaling is one of the main antiviral or main transcription factors triggering antiviral signaling, it basically hints at a facilitated or at an easier antiviral response in Omicron bystander cells that we don't see in the other two variants. So we wanted to locate those correlated genes and again check the data in a spatial context. And you see here again that all these reddish dots positioned in the whitish areas represent NF-κB inhibitor alpha, hence highly infected cells. But we also see two areas of infected cells that respond quite differently. So in area 1, here on the right top image, you see highly infected cells marked by red NF-κB colored particles. And you also see in the middle a cell that is expressing interleukin-6, shown in blue, and CXCL8, shown in yellow, that is not yet highly infected. So this cell might represent a bystander cell that is responding to beginning infection or to paracrine signaling. At the bottom right image, you see also infected area that does not respond to the infection at all. It seems that the virus to some degree managed immune evasion, or that the cells simply did not yet realize that they are infected, or that it really takes a larger amount of viral particles for the cells to really, or for the virus to induce NF-κB inhibitor alpha expression and to mount antiviral signaling and so on. So especially this cell marked by arrows in the bottom is really interesting, because you still see individual SARS-CoV-2 particles marked by whitish dots in the cell. That indicates an emerging infection. So yeah, I wanted to point out the severe heterogeneity in the cell culture again. So it really depends on which cells you look at precisely in order to make sense of the expression data that you get. So here are the images from Delta and Omicron with the correlated genes. In green are again marked the antiviral genes. And I want to draw your attention to C here and the enlarged image inset. You see a highly infected cell marked by the white frame, and you have another cell just below the highly infected cell that is not infected yet expresses a large number of antiviral genes. And this cell is also, or we hypothesize that this cell is showing an increased antiviral response due to paracrine signaling, because it's directly neighboring the highly infected cell, and thus might be mounting an antiviral state. But if you look at the larger picture, you see that there is not many of these cells that are not infected yet mount an antiviral response. So here it's really about how the cells are able to induce antiviral signaling, innate immune responses, and to inform other neighboring cells on their infection. So in D, we have the same images from Omicron. And here I want to mention that Omicron has an increasing rate of correlated antiviral genes. So here we have already 6 antiviral genes that relate in Omicron, but only 5 in Delta and 4 that related in Wuhan. So we kind of see that in Omicron, in bystander cells as well, we have an increased antiviral response compared to the other 2 variants. I shortly want to come back to the spatial context that is really important for us here. So if you look at the image at the top left corner, we see 2 infected cells marked by the white frames and 1 cell kind of in between these cells that actually shows the antiviral response, with genes marked in green and in bluish, which represent the antiviral genes in SARS-CoV-2. If you look at the picture, the middle picture on the left side, you see that it's actually again the bystander cell that produces or responds with the antiviral response, again in green and bluish, and not the highly infected cell shown in white, which is framed with the black frame. And we see the very same in Omicron in the bottom image, where we have the highly infected cell with the whitish area that shows some kind of antiviral response, but not as strong as its neighboring cell. So if we move forward to some statistics overall analysis, we see that it's actually C19, ORF66, and OS1 that are indeed higher expressed in bystander cells than they are in infected cells. So at this point, we strongly think or hypothesize that it is these 2 genes in bystander cells that potentially make a difference between how well the virus spreads in the culture. Something very interesting was that we found that PDCD1 is also higher expressed in bystander cells in Delta infected cells than it is in infected cells in the same culture or in cells infected with other variants. And PDCD1 has important implication in immunotherapy for immune evasion. So we are also looking into whether this gene actually has some kind of, whether it's important for actually immune evasion in SARS-CoV-2 infections. So using the spatial context that we are investigating, we checked whether genes or whether the virus particles that infect a primary cell also, well, let's say whether genes that the virus particles that are expressed and released from the primary infected cell induce antiviral responses in areas surrounding that cell in a well, space-dependent context. And we investigated several infection sites. We divided these into different regions of interest and checked the expression rates of different genes across these regions of interest. And by checking which genes are up or down regulated in specific regions of interest, we can kind of guess how far the viral spread has already advanced in that infection site. What we found is that if you look at the left quadrant of region of interest 1, we see that genes upregulated, which are represented in quadrant 1 here and marked in red, kind of tend to become downregulated in region of interest 4 here on the right side. And this is the contrary way for genes initially downregulated in region of interest 1, which seems to become upregulated in region of interest 4. So there might be some kind of systemic effect as well of these genes in these different regions of interest. So this is actually a very nice example on how the spatial analysis can actually be used to track virus spread and virus antiviral signaling throughout different regions of interest. So with that, I'm at the end of my talk, and I would like a lot of people, first of all, Professor Kurt Sadlocker, who leads all these investigations, and my team, especially Esther Melina, that helped me a lot with the experiments. And I also want to thank Resolve Biosciences, and especially Christian and Matthias, who also helped a lot with, helped us with analysis and also the different viral, well, there is the molecular cartography part of the entire investigation. So thanks a lot for all these people, and of course for you for listening. Thank you.